Welcome to our free learning university, Next Generation. Today, we are diving into one of the most electrifying scientific stories of all time, the race to uncover the secret of life itself. A mystery hidden inside every single cell of your body. A twisting code that decides who we are, what we look like, and even how we feel. This isn't just a story of science, it's a story of obsession, rivalry, genius, and the unstoppable human desire to know what makes life alive. This is the Code of Life. James Watson. Francis Crick and the DNA discovery. The mystery begins in the early 20th century. Biology stood at a crossroads. Scientists knew that something invisible inside our cells carried the blueprint of life. Traits like eye color, height, and even intelligence were passed from parents to children. But nobody knew what that something really was. They called it the hereditary material, a mysterious substance buried deep inside the nucleus of cells. Some believed it was proteins, with their complex structures and endless variety. Others whispered about nucleic acids, but most dismissed them as too simple to hold the complexity of life. And yet, hidden in that simple molecule was the most profound secret nature had ever written. In 1944, a quiet experiment began to shake the scientific world. Oswald Avery, an unassuming Canadian-American scientist, made a stunning discovery. He found that the molecule responsible for passing on genetic traits was deoxyribonucleic acid, or DNA, but the scientific community wasn't ready to believe it. How could something so chemically boring, just sugar, phosphate, and a few bases carry the entire design of a human being? Still, Avery's discovery planted a seed, one that would soon grow into one of the greatest races in scientific history. By the early 1950s, this race was heating up across the world. Laboratories buzzed with excitement and competition at King's College, London. A brilliant but underappreciated scientist named Rosalind Franklin worked tirelessly with X-ray crystallography, a complex imaging technique that could reveal the hidden shapes of molecules. Her photographs were among the clearest ever taken of DNA, each blurry. Glowing X was a step closer to unveiling the structure that carried life's information. But Franklin's brilliance often went unrecognized. Her meticulous precision and quiet dedication made her both admired and isolated in a field dominated by egos and impatience. Meanwhile, across town at Cambridge University, two ambitious scientists, James Watson, a 23-year-old American with an infectious curiosity, and Francis Crick, a 35-year-old British physicist turned biologist, were growing restless. They wanted to crack the structure of DNA first, not just to understand it, but to own the discovery. Watson, energetic and bold, had an instinct for shortcuts and wild ideas. Crick, older and deeply analytical, had a mind like a machine, able to see patterns others missed. Together, they were a dangerous combination, part reckless, part genius. Inside the Cavendish laboratory, their desks overflowed with notes, wire models, and half-built molecular skeletons. They knew that DNA was made of 4 chemical bases: adenine, a thymine T, cytosine C, and guanine G. But how these pieces fit together was the ultimate mystery. Crick would say, "If we can figure out how these parts connect, we can understand how life copies itself." Watson grinned. "Then let's build it." They weren't just reading about DNA, they were trying to see it. At first, their models were wrong. The early versions of their DNA structures twisted the molecule the wrong way, stacked the bases incorrectly, and ignored key data from other researchers. The Cavendish head, Sir Lawrence Bragg, even told them to stop working on DNA altogether. But Watson and Crick refused to quit. Their secret weapon wasn't just imagination, it was their obsession with the work of others. They borrowed insights, compared notes, and pieced together every clue they could find. And then came the moment that changed everything. One day, Watson visited King's College and saw something that stopped him cold: a photograph taken by Rosalind Franklin. It was labeled Photo 51. It looked like a simple X-ray diffraction pattern, a blur of lines and spots. But to Watson's trained eye, it was something else entirely: a spiral, a helix. He rushed back to Crick, eyes wide with excitement. "Francis, it's a helix. The bases must be stacked inside like steps on a spiral staircase." Crick's mind exploded with possibilities. "That's it. It has to twist. Two strands coiling around each other, and the bases on the inside must pair specifically." They began to build, wire and metal rods forming the elegant double structure that would soon become one of the most iconic images in science. Each piece clicked into place: phosphate on the outside, bases on the inside. And then came the realization that made everything fit perfectly. Adenine always paired with thymine, and cytosine always paired with guanine, two strands perfectly matched, capable of copying themselves, a molecule that could replicate, evolve, and store the code of life. Watson leaned back, eyes gleaming. "This," he said quietly, "is the secret of life." Crick smiled. "And we've found it." On February 28, 1953, the model was complete, a shimmering double helix standing tall in the lab. The two scientists ran to a nearby pub, breathless with excitement. Crick raised his glass and announced to everyone, "We have discovered the secret of life." The patrons, unaware of what had just happened, simply clapped politely. But in that moment, the world changed forever. Watson and Crick had decoded the structure that made evolution possible. Heredity real and biology eternal, far away. Rosalind Franklin continued her quiet research, unaware that her photograph had been the missing piece in someone else's puzzle. She would never fully receive credit in her lifetime. But her precision, her intellect, and her courage had been as vital as any wire or equation in the Cavendish lab. And so began a new era. The discovery of DNA's double helix was not just a victory for science, it was a revolution for humanity. It gave birth to molecular biology, genetics, cloning, and even the possibility of creating life itself. But the journey was only beginning. Because understanding the shape of life was one thing, learning to control it would be something else entirely. The revolution unfolds. The discovery of the DNA double helix was more than a scientific triumph. It was a spark that ignited the modern age of biology. When James Watson and Francis Crick built their elegant model in the Cavendish laboratory, they didn't just reveal how life copies itself, they opened a door to understanding everything about what makes us who we are. News of their discovery spread like wildfire through the scientific world. Within days, whispers turned to headlines, and laboratories from Cambridge to California were buzzing with a single question: if we can read the code of life, can we change it? But behind the scenes, the path from discovery to recognition was messy, emotional, and deeply human. At King's College, Rosalind Franklin continued her research with quiet focus. She had taken the X-ray photograph, Photo 51, that had given Watson and Crick the final clue they needed. But she didn't know that her data had been shown to them without her explicit permission. The truth was complicated. Maurice Wilkins, Franklin's colleague, had shared the image with Watson, thinking the research was collaborative. But Franklin saw things differently. She valued accuracy over speed. And she believed in understanding every detail before announcing a grand claim, while the men at Cambridge raced ahead. She was methodically refining her own models, inching ever closer to the same answer. In the spring of 1953, Watson and Crick submitted their paper to the journal Nature. Their words were simple, but world-changing. "We wish to suggest a structure for the salt of deoxyribonucleic acid DNA. This structure has novel features which are of considerable biological interest." On April 25, 1953, Nature published not one, but 3 papers on DNA. Franklin's and Wilkins's papers appeared, alongside Watson and Crick's, a scientific puzzle, completed from 3 different angles. But history would remember only 2 names at Cambridge. Crick celebrated with uncontainable joy. He believed their discovery was the single most significant event in biology since Darwin's Origin of Species. Watson, younger and restless, was already thinking about what came next, how to decode the genetic message itself. If DNA carried instructions, what was the language? How did it tell cells to build muscles, brains, and bones? Meanwhile, Rosalind Franklin had moved on to Birkbeck College, where she began studying viruses, tiny packets of genetic material wrapped in protein. Her work would later help define the structure of the tobacco mosaic virus, laying the foundation for future virology. Tragically, in 1958, Franklin's promising career was cut short by ovarian cancer. She was only 37. Her notebooks, filled with precise calculations and X-ray images, became silent witnesses to one of science's greatest unsung contributions. While the scientific community mourned Franklin's loss, Watson and Crick's fame grew. In 1962, they, along with Maurice Wilkins, were awarded the Nobel Prize in Physiology or Medicine for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. It was a victory, but also a controversy. Many argued that Franklin deserved equal recognition. Yet Nobel Prizes are not awarded posthumously, and so her name was left out of the official record. Still, history would not forget her. Decades later, her work would be re-examined, and she would emerge not as a footnote, but as a vital part of the greatest biological discovery of the 20th century. After 1962, the double helix began to transform from a symbol of discovery to a tool of innovation. Across the globe, young scientists started dreaming bigger. What if we could read the entire DNA sequence? What if we could rewrite it? The 1970s saw the rise of genetic engineering, the manipulation of DNA, to alter the genetic code of living organisms. Suddenly, the building blocks of life were no longer untouchable mysteries. They were editable instructions. Laboratories learned to splice genes from one organism into another. The first recombinant DNA experiments were performed, and by 1973, scientists had inserted a frog gene into a bacterium. It was both thrilling and terrifying. For the first time, humans had the power to alter the natural order to create lifeforms that had never existed before. The discovery of DNA structure had not only revolutionized biology, it had blurred the line between discovery and creation. Philosophers, politicians, and religious leaders began to ask hard questions. If we can modify life, where do we draw the line? Can science move faster than morality? In 1975, a group of the world's leading scientists gathered in Asilomar, California, to discuss the dangers and ethics of genetic manipulation. The Asilomar Conference became one of the most important meetings in scientific history, a moment when scientists paused to consider the weight of their own power. James Watson was there, older now, but still bold. He believed that the benefits of understanding DNA far outweighed the risks. If we fear knowledge, he said, we give ignorance power. By the 1980s, DNA had become the foundation of an entirely new field, biotechnology. Companies began using genetic engineering to create synthetic insulin, saving millions of diabetic lives. In 1983, Kary Mullis invented PCR polymerase chain reaction, a technique that could copy DNA rapidly and endlessly. Suddenly, genetic analysis became faster, cheaper, and more precise. The helix that Watson and Crick had built in wire and metal had now become a code in silicon and software. The bridge between biology and technology. And yet, through all of this progress, one haunting question remained. If DNA is the code of life, who wrote the code? For Watson and Crick, the discovery had been about how, not why. But for millions around the world, the double helix seemed to whisper something deeper. That life was written in a universal language, shared by every living thing on Earth. It was elegant. It was mathematical. It was beautiful, and for the first time in human history, we could read it in the years that followed. The double helix became more than just a molecule. It became a cultural icon. Artists painted it. Movies used it. Even jewelry designers shaped it into silver spirals. DNA was now a symbol of identity, evolution, and destiny. Yet, as we celebrated this triumph, scientists knew the story wasn't over. Understanding the structure was just step one. Now came the ultimate challenge: decoding the entire genetic blueprint of humanity. And that next chapter, the Human Genome Project, would test not just our intelligence, but our ethics, our unity, and our idea of what it means to be human. The code of life had been revealed, but its message was only beginning to unfold. The human blueprint. The year was 1984, and a quiet revolution was about to begin. In laboratories from London to California, a small group of scientists gathered to discuss a dream that seemed almost impossible: to read every single letter of the human genetic code. Not just parts of it, not just genes for diseases or traits, but all of it. This dream would become known as the Human Genome Project. The most ambitious scientific effort since the moon landing. At its heart was a simple but staggering question. What if we could read the entire instruction manual of human life? Each person's DNA contained more than 3 billion chemical letters, a T, C, and G spiraling together in that elegant double helix. These letters formed the blueprint for every cell, every heartbeat, every thought. But deciphering that blueprint was like trying to read a library written in a language no one yet understood. And yet, the scientists were determined to try. In the early meetings, tensions ran high. Some thought the project was too big, too expensive, too ambitious. "It's madness," one critic said. It would take a century to complete. But a few visionaries, people inspired by Watson and Crick's discovery, believed it could be done. Among them was James Watson himself. Now, older, wiser, and deeply influential, he saw the genome as the next logical step in humanity's quest for self-knowledge. "We discovered the structure," he said. "Now we must read the code." In 1990, the U.S. Department of Energy and the National Institutes of Health officially launched the Human Genome Project. It was a global collaboration involving scientists from the United States, the United Kingdom, Japan, France, Germany, and China. Their goal: to identify all the approximately 20,000 to 25,000 human genes and map the entire sequence of the 3 billion DNA base pairs that make up the human genome. It was science at its grandest, and most personal to the public. It sounded like science fiction. Reading DNA, mapping humanity. Was this even possible? But inside the laboratories, the atmosphere was electric. Massive DNA sequencers hummed day and night, spitting out fragments of the code. Technicians ran gels, computer scientists wrote algorithms, and biologists pored over patterns for the first time. Biology had become data science. The double helix was no longer just a molecule. It was information. And information meant power. As the project gained momentum, something extraordinary began to happen. Competition. A private company called Celera Genomics, led by the bold and controversial scientist Craig Venter, announced it would sequence the entire human genome faster and cheaper than the government-backed project. The race was on. Watson, who had once been the rebel scientist himself, now watched from the sidelines as 2 teams sprinted toward the finish line. The tension between collaboration and competition, between open science and corporate secrecy, echoed the same conflicts that had surrounded DNA's discovery decades earlier. Venter's Celera team used a new technique called shotgun sequencing, breaking DNA into random fragments, sequencing them rapidly, and using powerful computers to reassemble the puzzle. The public consortium, meanwhile, used a slower but more methodical approach, sequencing one chromosome at a time. The world watched as science turned into a high-stakes race. Then, on June 26, 2000, in a moment that would be replayed across every news channel on Earth, two men stood side by side in the White House: Bill Clinton, the President of the United States, and Tony Blair, the Prime Minister of the United Kingdom. Behind them were Francis Collins, head of the Human Genome Project, and Craig Venter, representing Celera Genomics. Clinton began his speech with words that captured the magnitude of the moment. Today, we are learning the language in which God created life. The crowd erupted in applause. Humanity had just completed a working draft of its own instruction manual, the blueprint of life. Itself, it was a breathtaking moment. But it came with new questions, profound, unsettling questions. If we can read our genetic code, can we predict our future? Can we edit out disease? Can we design intelligence, beauty, or strength? The line between science and philosophy began to blur once again. Watson, ever the provocateur, warned that knowledge without compassion could be dangerous. He believed in the pursuit of truth, but also in the humility to respect life's complexity. Francis Collins, a geneticist and a man of deep faith, spoke about the moral responsibility of understanding the genome. "We are not playing God," he said. "We are learning how he works." Over the next few years, the Human Genome Project was refined and completed. By 2003, the full sequence of the human genome was officially published 50 years to the day after. Watson and Crick's paper on the structure of DNA, the symmetry was almost poetic. The double helix had come full circle. But this was not an ending. It was a beginning, the dawn of genomic medicine. Doctors could now identify genetic mutations that cause diseases like cystic fibrosis, Huntington's, and sickle cell anemia. For the first time, medicine could target the cause, not just the symptoms. Pharmaceutical companies began developing drugs that worked with specific genetic profiles. A new era of personalized medicine was being born. And yet, amid the excitement, scientists wrestled with the ethics of it all. Should genetic data be public or private? Could employers or insurance companies use it to discriminate? Was humanity ready to hold its own blueprint in its hands? Meanwhile, a new generation of scientists began to dream even bigger. The genome had been mapped. Now they wanted to rewrite it. From the halls of MIT to labs in Tokyo, researchers began experimenting with gene editing. The idea was both simple and terrifying. Use a molecular tool to cut and modify DNA with precision in 2012. To women, Jennifer Doudna and Emmanuel Charpentier introduced a breakthrough technology called CRISPR-Cas9, a tool that could edit genes quickly, cheaply, and accurately. It was the most powerful invention since the double helix itself. Now, humans could not only read the code of life, they could edit it. The implications were staggering. Scientists began correcting genetic diseases in embryos, plants, and animals. Others imagined erasing hereditary conditions entirely. Some dreamed of extending human life, even designing future generations. But others warned that tampering with life's code was a step into the unknown. Would we still be human if we rewrote what it means to be human? In classrooms around the world, children were now learning about DNA not as a mysterious spiral in a textbook, but as a living code inside them. DNA ancestry kits became popular. People spat into tubes to discover where they came from, Africa, Europe, Asia, all connected by a single shared code. The double helix had become a symbol of unity. Proof that beneath our differences, we were all part of the same genetic story. And somewhere in that spiral of A's, T's, C's, and G's lay the instructions for everything we are and everything we might become. Rewriting the future. The age of gene editing, the discovery of CRISPR-Cas9 in 2012, marked a turning point, not just in science, but in the story of life itself. What Watson and Crick had uncovered in 1953 as the structure of DNA, humanity now held the power to rewrite. The double helix was no longer sacred text. It was editable code in a quiet laboratory at the University of California. Jennifer Doudna sat before a computer model of the CRISPR system, tracing the elegant mechanism by which bacteria had evolved to defend themselves against viruses. It was brilliant, simple yet unstoppable. Nature itself had built a pair of genetic scissors, and now humans could wield them. Imagine being able to cut and paste DNA as easily as editing text, she said. That single sentence sent shockwaves through the scientific community. For centuries, biology had been descriptive, scientists observed, recorded, and classified. Now it was creative. CRISPR gave humanity the power to design. Within months, labs across the world began experimenting. Scientists corrected defective genes in mice, engineered plants resistant to drought, and even attempted to remove HIV from infected cells. The early results were staggering, like stepping into a new era of creation. But as always, power comes with questions. If we could fix broken genes, could we also enhance the healthy ones? If we could remove disease, could we also choose intelligence, height, or appearance? The idea of the designer baby quickly entered headlines, igniting fierce debates. Some saw it as the future of evolution, the ability to eliminate suffering. Others saw it as the end of natural humanity. At the 2015 International Summit on Human Gene Editing in Washington, D.C., the world's leading scientists, ethicists, and philosophers gathered to decide one thing. Should humans ever edit their own germline DNA, the code passed to future generations? The room was silent as Doudna spoke. "We must proceed with caution," she said softly, her voice echoing through the hall. "We have opened a door that can never be closed," but the door was already open in 2018. A Chinese scientist named He Jiankui stunned the world when he announced that he had created the first genetically edited babies, twin girls whose DNA had been modified to resist HIV infection. The world erupted in outrage. Scientists condemned his actions as reckless and unethical. Governments banned human germline editing. He Jiankui was arrested, his career destroyed. But the message was clear. Humanity had crossed the threshold. The era of gene editing had begun. Behind the chaos, technology continued to evolve. AI and machine learning entered genetics, decoding patterns faster than ever before. What once took years could now be done in hours. Algorithms began predicting how specific edits might affect physical traits or disease. Resistance, the fusion of AI and biology, transformed science into something almost godlike. In one corner of a glowing laboratory, a robotic arm moved with perfect precision, extracting strands of DNA from samples. On the screens, colorful double helices danced, each representing a person's genetic code. The scientists didn't need to touch the data. The AI did it all, reading, comparing, learning what Watson and Crick had drawn by hand in 1953. AI could now model in seconds billions of bases at once, in full color, alive and breathing on digital screens. Meanwhile, in hospitals, the effects of gene editing began changing lives. Children born with fatal diseases like spinal muscular atrophy or sickle cell anemia received treatments that repaired their faulty genes. Conditions once considered incurable began disappearing. Parents wept as doctors told them their children would live normal lives. The impossible had become possible. Yet in the shadows, a new anxiety grew. If we could edit our flaws, would we still value imperfection? If we could create perfection, what would happen to humanity's soul? Its chaos, its creativity, its empathy. One night, Francis Collins, now older and reflecting on decades of genetic work, gave a speech at the National Institutes of Health. His voice carried the weight of history. When Watson and Crick revealed DNA, he said, we glimpsed the language of life. When we sequenced the genome, we began to read it. But now we're rewriting it. And I wonder who will be the author of humanity's next chapter. The audience sat in silence. By the mid-2020s, CRISPR had advanced even further. Scientists developed base editors tools, so precise they could change a single letter in the genetic code without cutting the DNA strand. It was like correcting a typo in a novel without damaging the page. Other teams worked on prime editing, allowing near-perfect accuracy in rewriting sequences. Gene therapy entered the mainstream. DNA editing kits became cheaper, smaller, and available to universities worldwide. The once impossible dream of controlling evolution was now reality at the same time. Artists, philosophers, and activists began asking deeper questions. If our genes define us, what happens to identity when genes are rewritten? Would a genetically edited person still feel human, or would they be something new? Documentaries, books, and films began exploring these questions. The image of the double helix appeared on billboards, tattoos, and protest signs. It had become the most recognizable symbol in science, and perhaps the most misunderstood. In schools, children learned how DNA editing worked using colorful animations. In science museums, visitors watched holographic models of CRISPR in action. The mystery that had once belonged only to laboratories now belonged to the public. And yet, one truth remained the same truth that had driven Watson and Crick, Rosalind Franklin, and all those who followed: curiosity. It was curiosity that drove humans to split the atom, to walk on the moon, and now to rewrite life in the quiet of her lab. Jennifer Doudna once had a nightmare, a dream in which someone asked her to show them how CRISPR worked. She explained, and the person smiled. But when she looked closer, it wasn't a scientist, it was Adolf Hitler. She woke up shaken. That dream haunted her. It reminded her that every scientific discovery carries both promise and peril. Humanity had always danced on the edge of its own brilliance. By the end of the decade, the double helix had evolved from a symbol of discovery to a symbol of power. DNA was not just biology anymore. It was identity, data, and destiny. Humanity stood at a crossroads. One path led to healing and enlightenment. The other to control and inequality. Would the code of life become our greatest gift or our greatest temptation?